Johanna, thank you very much for joining me to talk a bit about the image analysis at Vibraint and the CRO service module. Could you start off by telling me a bit about your background and what you do at Vibraint?
Hello. Yes. My name is Johanna, and I'm co-founder and CTO at Vibraint right now. I have actually a background in physics, especially biophysics and computer science. I studied in Technical University of Munich, and then I have been also in Stockholm, and then now in Denmark. So I have come through many different countries, and so at first. I was very, very excited about all the different kinds of imaging methods. So in my physics specialty, I actually focused a lot on MRI, for example, and different
Interesting.
fluorescence microscopy and spectroscopy techniques. And I wanted to know all about it. And at one point I realized that because if you're in the physics field, you're expected that you kind of start developing these techniques and the hardware and so on. I realized that these technologies are, quite advanced already, and that the bottleneck is actually more in a signal processing side. So I did my master's thesis in Karolinska Institute on functional MRI, and from there I moved also then to light sheet microscopy. They're both, so I both in both cases worked with whole brains, whole mouse brain imaging, so to say, and I got really excited about brain image analysis basically. Yes, I then after my PhD, I did an industrial PhD in a biotech company called Gubra, and also Technical University of Denmark, which was then focused on actually combining MRI and light sheet microscopy. So I built like a multimodal brain atlas because at one point there was this realization that after doing MRI and after doing light sheet, that all these worlds are very separate. It's and actually they all provide complementary information. And that is where all this integration of different 3D brain imaging modalities actually came into play. And that's also why, in the end, Vibraint was born together with Jakob and Thomas.
Yeah, do you want to tell me a little bit about how Vibraint was born, and I guess to the gap that you identified that you wanted to address?
Yeah, I think the gap exactly was that the problem is that there are so many good imaging technologies out there, and they're complementary information. They're all valuable, from in vivo imaging to ex vivo microscope imaging to then even spatial transcriptomics, and they kind of these worlds live in their own spaces. They all provide information, but it's extremely difficult to combine it all. And we all actually, Jakob and Thomas and me, we all had that feeling that this, to actually make sense of this data, you need to start looking at it in a combined and integrated format. And this is the reason we basically built Vibraint, and that is also our aim, because we feel that neuroscience has the research has been stagnated a little bit, and everyone are trying to combine these techniques, but it's just very, very difficult. So we're trying to build tools and have then services around that to actually help people to combine data.
So you've given a bit of a taste there of a bit of a hint as to what you guys actually offer there, this kind of integration of pulling all of this stuff together. But what is your sort of elevator pitch for Vibraint and the service that you offer then?
Mm-hmm. We are, we identify ourselves as a deep tech company for preclinical neuroscience. So as you said, we have offerings in contract research part, but we also develop software tools, which we then either use in our services, but we also offer it as a standalone tool, so to say. So in our services, in the contract service side, we offer a modular pipeline, meaning that it's possible to order modules of, for example, tissue clearing, so tissue processing, from staining to clearing, then the imaging of whole brain samples, and then image analysis of the whole brain samples, which then actually bridges to our brain viewer, where we bring all our results together in one web-based application, basically.
Fantastic. Thank you very much. Building upon that, where do you fit into this picture as CTO? Like, what's your role within Vibraint?
Yes, I'm responsible for technology development. So meaning as an expert in image analysis, I'm developing the platform for image analysis, but then also the brain viewer software that we have in the web. Yes, exactly. So I'm overseeing these two product lines, you could say. Exactly. And just to maybe emphasize here is that because our product line in the contract services is modular, then we are really happy to also support different laboratories in analyzing their samples that they have acquired in-house. We have seen that this is a quite important point because light sheet microscopy has now been around for sure more than 6-7 years, I think even 10 years. And in the beginning, it was, I need to say, the bottleneck was acquisition of data. Then now, many laboratories actually can afford to have the microscope in-house. Many companies have it, their core facilities have these, even like standalone laboratories, research departments, and universities. But people really struggle with analysis of these samples. And that's because we have been in that world for a long time. So all of us, Thomas and Jakob and me, and we have all the expertise in how to do it because that's what we have been doing for a long time now. We saw opportunity to help people because not every institution has image analysts or then image analysts dedicated for brain analysis.
That was actually you've actually. I was going to ask you exactly about that. So yeah, not all labs or institutions can do this kind of stuff in-house, essentially.
Yes, exactly. There are obviously institutions and laboratories that have computational scientists in-house. Very often they need to do analysis for many different kind of techniques, many different kind of organs. And we believe that brain is a very complex organ. It also there are certain kind of analysis steps you need to do to analyze the samples, which means that it's maybe for a computer scientist that is, or data analyst who are supporting everyone in the facility, it's just very difficult to know exactly how this is done. And people need to learn almost the whole field to be able to do it.
So that, would you say that's what makes the analysis aspect of light sheet data so difficult?
Yes, that's one part of it. So there are several aspects. One of it is also that the data is really big. Data sets, I mean, usually if you image one brain, nowadays these microscopes can generate a lot of data. So within 1 hour, you can for sure scan even 1-2 brains nowadays. And one brain sample with not even very, very high resolution is, or to have good enough resolution, for example, 5 micron isotropic, and then you usually acquire several channels, it's easily 20 or 40 gigabytes. And then if you go higher in resolution, then obviously it's easily can reach up to several hundreds of gigabytes, up to terabyte in the case of rat brain, for example. So it's a lot of data. So that's one challenge. So you need dedicated hardware, dedicated techniques, how to handle these big data sets. The other side of it is like I mentioned, that brain is, there are opportunities to analyze it more in a different manners than you would do with other organs, for example. You can use brain atlases because brain structures are very conserved within animals of the same species, of the same strain. And by using atlases, you can start doing, because you can map brains into same space, you can start doing different kind of analysis than you would otherwise do with other organs. And what I also think is a difficulty, actually, is I see it as an interdisciplinary field, especially because image analysts are very often computer scientists or mathematicians, because you need like this mathematical logic, how you would analyze images, or you use mathematical methods to do that. But this doesn't mean that you understand the neurobiology, for example, or because every single step in this pipeline really matters. I also see this as a interdisciplinary field, and you need knowledge and skills from different fields, from neurobiology to imaging, actually imaging physics, even where does the signal come from, really? What is it? To really understand what you measure, but also what artifacts you have. And then you need the person who is able to also perform the mathematical algorithms on the image to extract information. So this having a team of people who have worked together very closely and know how much every single step affects what comes out in the end, this is really important. And I think this is why it is so important to offer also image analysis as a standalone service from our side, because we have that team and we have gathered the knowledge over years, so to say.
Yeah. That's a good answer, because I was going to ask you then, where do you guys fit into addressing some of those challenges? So for example, like your team expertise there, and offering this as a bolt-on rather than a sort of comprehensive end-to-end package.
So we do both, because sometimes research groups, very often they focus on certain techniques. Actually, they have their go-to techniques that they're really good at, and then they want to supplement their findings or validate them using a different technique. Or it's because whole brain technology is also a little bit for it's exploratory also in the same time, to first find interesting places in brain and then go in depth. So what I want to say with that is that very often it's a technique that doesn't need to be part of a typical method package that the laboratory has, or like a research group has, because it's one-off, or they do it very seldomly. Then they have opportunity to buy the full package. Because we know how to do tissue stainings and clearings for whole mount brains, how to image them and how to analyze. But if people prefer to do certain sets themselves, this is also then possible, because we understand that people might be skilled in different areas and might have different needs depending on who is currently in their lab available. Yeah.
You've listed some really good reasons there as to why you, as a lab, you might want to take on this service, but what would you say to people who are on the fence about whether to go with you or maybe try and to do this internally?
I think the biggest benefit is first of all, that if you, for example, employ a PhD or postdoc on a project, and part of it is to do light sheet microscopy, very often it takes long time to optimize these protocols. That's sadly the reality. We actually do provide the protocols for free and make them available for people so that they're able to do them in-house without spending months and months and months optimizing them. Because one thing is, I mean, tissue quality has a very big impact. Also, imaging quality has a very high impact. So you need to first reach the level where you actually have good data to analyze. But we're very happy to support laboratories with as much knowledge that we have, gathered over the years, optimizing many antibodies, clearing techniques, and so on. So we would like that they can speed up their research. The problem is that otherwise a PhD student or postdoc might spend their whole time only trying to get the light sheet data out, and then they still need to analyze it, which they don't know how to do. And so we're supporting with information, but also we also consult people if they like to do their own analysis. But I think the biggest point is speeding up. And then the other part is also the quality that we can provide, because we have been running high throughput pipelines for a long time, and we know that our output is reliable and also comparable to other data sets out there.
So my takeaway from that is that it's a bidirectional relationship as well. It's not just like they gather their data or do the full pipeline and they just send it to you and you process it and give it back to them. There's more to it than that. You're giving back into the system in more than just processing the data.
Yes, exactly. We would like to be part of the process, and also just to mention that if we are analyzing or helping to analyze their data, it's actually very important to be in the loop a little bit earlier, before even the data is actually acquired, because the way how the data is acquired depends a lot what you can extract, what kind of information. So before even starting this whole experiment, you need to know what you want to get out of it, to then know what settings to use for imaging. And then there is a certain set of information that you can gather then from the images. Because every time you do a decision, it's a trade-off on something. So it's very important to know, because it's difficult. It might be difficult to analyze data sets that have just been acquired without keeping in mind what they want to get. Like, what is the quantitative measure they want to actually compare between study groups or analyze? Yeah.
No, that's really interesting. And feel free to go rather technical on this question, but how flexible are you with that sort of stage of the pipeline if you do come in after, they've made the decisions on how they're gathering the data? How flexible are you working with that kind of thing?
Yeah, as I mentioned, I think we try to be flexible, and I think one part of it is that we curate the data, and we need to see, is this quality good enough, or is this image taken acquired the way that it is possible to extract information that they would like to. Because there are many different endpoints you can measure. You can measure intensities, or you can measure the signal coverage, or you can measure every single, for example, nuclei or cell separately, count them. But they all are meaningful for certain biological questions. But in the same time, they also come with their own challenges. If researchers have used wrong resolution to image, or even step size in Z, it might be impossible to actually detect every single object and separate the objects that cluster.
Interesting. So do you want to tell me a bit about the setup and the tools that you guys are using for performing the analysis?
Yes, we do image analysis on site. We use our own servers for doing that. And all the analysis is actually done on Python coded pipelines. We are also right now looking into putting data sets, or like putting image analysis also into cloud, but right now it's fully done on site. And we don't use any commercial other softwares right now, so it's fully coded by us. Yes.
And how do you, working with that in-house, how do you were talking about the data earlier on, about how big these data sets are. How are you managing that data internally?
Exactly. So this goes a little bit into this experimental planning side, and knowing what you really want to measure, what is relevant also to measure in your biological experiment. So first up actually starts from there, to understand what is a resolution that you need. So we always try to use the lowest resolution possible, obviously, to be able to analyze them. And we do not do too many data transfers. So we try to optimize every movement, and that we use dedicated hardware for every step. We use also image compression quite a lot to save space. And for example, for performing analysis of these huge data sets, you usually need to analyze, or at least, for example, for segmentation purposes, you need to load in only subvolumes or subchunks that you then later on need to stitch together in an optimal manner without creating artifacts. So there is, at every step, there is also a curation that everything has gone correctly. So for already when data is produced, the very first step is actually to do quality control, to see if this resolution is enough. Is the data set high quality? And then for every single step in processing and analysis, we always perform quality control.
Got you. And then how, going from the raw scan to the outputs, or to the results, what's the full sort of workflow or process that goes on there?
Yes. So very first thing, because mostly these light sheet microscopes, they acquire data in files. So there is a part that nowadays actually quite often is already done by the software of the microscope. Is, for example, you need to stitch the tiles together. Then the downsampling is often applied to the resolution that you need. And then sometimes there's also additional post-processing steps that people might like to apply. For example, destriping, which would remove some striping artifacts. Or there's something called deconvolution, because in the C direction, there will be very often elongation of the signal. And there might be more post-processing steps. But very often, I try to be pragmatic and also say that the more operations you do on your image, the more you also modify the signal. So it's very important to be aware of it, why or what you're measuring, to know what kind of post-processing steps are okay to make. But I think the most important thing is always stitching. And then what we also do is that we orient the samples all in a standard position before they even enter our analysis pipeline, actually. And then the steps in the analysis pipeline, there are many layers. It's also actually a very modular structure. So one of the first things we do is brain registration to the atlas and atlas registration to the sample. So it goes both ways. But as I mentioned, because our brains are very conserved, we are able to use brain atlases to then define and delineate brain regions, which means that we can measure certain metrics inside these brain regions. And for that, we use atlases. And for that case, we need to, every single sample we have acquired, we need to align with the atlas. And also we need to align atlas to the sample because these are used for different cases. So from this process, we get something called transformation matrices. So we can move in both directions, either in sample space or atlas space. Then when we have done that, there is a very important step before even again starting this full-blown analysis, is that we need to find the segmentation technique we want to use. So how to extract the signal from different images. Either we might use classical algorithms that have been published, or it's possible to use AI models. And very often we try to then find the optimal algorithm for segmenting the signal.
And does that depend on what markers were used for the
exactly
sample?
Exactly, it depends on the molecular markers that were stained in images. So they are very often actually specific to these markers. But then for every marker, you might have different cases how to analyze or how to solve it. So it might either your AI models, you either might need to make AI models, or you need to fine tune them, or if you use classical models, you need to do parameter tuning. And you need to find a parameter that then fits all your samples that you have imaged. So that kind of all happens before the high throughput pipeline starts up, so to say. When we have found the optimal model or fine-tuned it or found the parameters for classical models, or very often it's also a combination of both, then we run the segmentation for all the samples. Then there is a quantification step where, for example, there are two kinds of analysis. So one of them is called region-wise analysis, and the other one is called voxel-wise analysis. Region-wise analysis is always done in sample space, meaning that now you would take your brain atlas regions, you will transform them on top of your sample, and then you would extract, using the segmentations, you would extract information from every single brain region. And then it depends what was your metric you decided to choose for analysis. Do you want to count something? Do you look at densities? Do you look at how far spread is a signal? There are many different metrics you can use, but then you would quantify that for every single brain region, so to say, and extract the number. And then you could move also into the voxel-wise analysis, where now the every sample. So actually, we extract in quantification step. We also extract segmentations or then objects that we have detected, and we can move them to a lower resolution space, which is the same resolution as the atlas, so that no information goes lost, but you can transfer it basically in the lower resolution space. And then you can map it to the atlas, because then there are very cool things that you can start making. You can start comparing animals to each other, also comparing groups, different study groups to each other, treatment groups. And you can start doing voxel-wise statistical analysis too. So we create then literally spatial p-value maps, but also average maps, or then combinations. Like, for example, you can look at which signals are common or differential between two study groups, for example. So there are these two paradigms, so to say, in analysis. Exactly. And then Also for region-wise analysis, we also perform then statistical testing, obviously. Yeah.
And then with all of this information, how is that surfaced? What does the final output look like here?
Exactly. So first of all, the brain maps that are either statistical maps, p-value maps, or then average heat maps that are dependent on, are you looking at cell counts or densities, coverage, or intensity? So it depends of that. So first of all, this is like brain maps is one thing. Then other thing is for region-wise analysis, you also get for every region the quantified metric. And obviously also variance within group and p-values between when you did the group comparisons, testing, and so on. And what we also, because the data, the problem with this data is quite complex. There is a lot of information. There is a lot of brain regions. There's more than 800. And if you think about voxels, there is even more, millions of voxels. The problem is how you look at the data. Like, how do you even explore it? How do you do it in an unbiased manner without cherry picking your regions? And for that, that was one of the reasons to also have the Brain Viewer and build tools to, in an unbiased manner, to actually browse your data. So that I think the unbiased here is a very important point. But what you can also do in a brain viewer, obviously you can put these, because all the brain maps that you have made, they're in Atlas space. But what you can then also look at is for every single individual brain, when you have detected objects, for example, if you look at nuclei, you can, or even cells, like if you have had a cytoplasmic stain, you can point them into in vivo coordinates in the Atlas space. And then if you click on every object, what you can start doing is that you can get also information for every single object. For example, what intensity or size, etc. it had, or how spherical it is, and so on. So you can extract a lot of information then for every single object also. So there is many different layers, and what the output is.
I think what you've really covered there is that is so much of the know-how that you were sort of explaining earlier in terms of what Vibraint can offer to laboratories or research groups that are looking to do this brain analysis, because there's so many parameters there. Even when you're talking about the resolution that you're gathering the images at, and I imagine you probably get some people who over compress the images that don't get enough detail, and that makes it difficult to do the analysis that you want to. And there's all of this post-processing that you're then doing as well, and you can see very clearly, like through that workflow, how much you guys have done this and how useful that must be to have that know-how.
Yes, I think there is a lot of details at every step. And I think also every algorithm you use, you might first think, oh, this is the perfect algorithm, I can use it forever. But then you might start, the more you understand the problems, the more you understand what actually, I think it's also evolution in the sense of, for example, what you even need to study when you study a certain model or certain staining. What is actually interesting there? Because very often scientists come and say, I want it all. I want all the metrics. I wanted everything. And then I wanted because what you can also do in every brain region, if you have all these metrics about sizes, intensities, etc. of the objects, you can start looking at distributions and compare distributions within brain regions. You can go more complex and more complex. And very often people want it all. But the problem is that very few of the metrics actually make sense. And it's an evolution of understanding what is a reasonable metric to evaluate. So I think this is also the experience that comes with the time. What is relevant now?
Yeah, and you can obviously help people with that. One of the interesting points that you raised as well, which I think is particularly valuable as well, is how you're doing the segmentation and analysis with a combination of classical but also deep learning models there. And you were talking about sort of training the model specifically to fit client's data. And I was interested to know a bit more about like how that works there, because I think it's a great point that you think you might have a model that you can apply to any brain analysis, but I imagine that fine tuning is really valuable. So I'd like to just know a little bit more about how you do that. Do you do that on an individual basis? So like what extent do you do this refinement? How does that kind of work?
Yes, working with AI models is a whole long procedure. Actually, it's also very I would say time consuming actually to create them, to maintain them, and so all this procedure is heavy procedure. However, I want to point out, and which is a little bit maybe funny to think, AI models have been around for a long time, and in computer vision, they have been used a long time. And it's so funny now to see how when LLMs are taking over the world, and then all computer vision scientists think, wow, but we're using them quite a while already. But what is cool with
Yeah,
them? The very nice thing actually with these models is exactly what you pointed out already a little bit, is that every time you acquire data, even in your own lab, the data will be different because your microscope will have, they also drift in time. So even if it's
firmware
same
updates,
microscope,
things like that.
exactly, you change a hardware piece, whatever, or your laser is just fading out a little bit, and so on. So even though you use same settings, you might get a different output. Or even if a person is doing tissue processing for years, every time there's a batch effect between different batches of samples. And now if you bring in the complexity that you have many samples and the complexity that there is actually many different laboratories and many different kind of microscopes that actually based on different architectures, how image is generated. So it's difficult. I need to say that, but in some other ways, it's very very motivating to try to think how to solve that problem. And I need to say, there are no foundational AI models right now. Nothing like this, except
Interesting.
for like light sheet data. I think people
Yeah,
go more and more in that direction, and we think about it.
yeah,
But the problem is every time you get your new experiment, you really need to evaluate how similar it is to the data before. And
yeah,
then if you have a model, you actually do need to fine tune it. So
yeah,
it's a procedure you need to do every single time, actually, at least to do a QC and then think, is this within my range of variance I'm okay with? But exactly. And I think that there are some stainings that are maybe more robust, where it is possible to create models that work very reliably, and also more and more the neuronal network architectures, get better. They also evolve, meaning that we will be better and better in detecting them. But I think the biggest problem is variability, but also variability not only between samples and batches, but also when you image them, the whole volume is, imaging settings are not always perfect in one end and another end. So there's a lot of variability. And the way we work with this is that all the data we have seen before, we actually try to, in a way, you could call it maybe mapping or clustering, trying to understand what are these possible signals and variances between signals that have ever occurred. And it's not like putting all the data just like into the model, because you need to
yeah.
always provide ground truth when you train a model.
Mm.
You actually start finding like representative, I would say, cluster centers then. So certain patches that represent a certain phenomenon, and then you include this into your training set and how you want that your AI model is handling it. So this is the training part. It's a lot of data, like going through data, curating it, understanding what's going on, and what is actually signal and what is not, because that's also a big question very often.
So could you simplify that almost as saying like you can take all of this data, feed it into a model that does almost like a summary or highlights that picks out these features that can be useful to then implement or fine tune into the other model that you're going to be
Yeah,
working with the data on?
exactly. So nowadays there are, for example, embedding models that you can use for describe patches, or so that it extracts different features. And then you can, for example, so embedding model is already an AI model, so you can then basically use different, and usually it's a combination of different techniques that you need to use. So in that case, for example, embeddings or also other characteristics of the patch are extracted with one model. And then you maybe use some further, maybe more classical techniques like clusterings to understand what kind of different types of signals you have, and then how you extract it. And then there's a question also, and that's obviously not so easy question, how much representative data of a certain phenomenon you also need to put into the model. So there are many questions
Yeah.
again. And these are, I would say, learnings every time, because you hardly ever know what is the problem. So it's kind of trial and error, trying out new things and seeing what it makes sense. But that's why I think the key is actually quality control always. And
Yeah.
I don't believe in these high throughput pipelines that don't do any QC, because if you don't check what comes out of your pipeline, I mean, you don't actually know if you found all the cells, if you know, you really don't know the status of if you can just use a model out of the box.
That's really interesting. There's so many points within that space there that I've got
Yes.
questions and things to touch upon. One of the key ones actually was when you mentioned about you have certain markers that you think perform better within for the whole analysis essentially. And is that a good example of where Vibraint coming in earlier in the pipeline, you can really help? Would you advise, for example, on those markers earlier on, or do you leave that to the labs? Will you provide input on that kind of thing?
I think we could provide information of which antibodies, for example, are reliable, or how to fluorescently mark the signal you're interested in. Definitely, yes. And because it takes a lot of optimization for every single staining to work, and some of them work better than others. And then definitely the know-how, what we have, we were really happy to share that. Exactly.
Yeah, and you raised a good point as well about AI as it's a difficult topic to bring up in sort of scientific conversations because of how popular it's become in sort of consumer technology. But that's why I refer to it as deep learning as well instead of AI, because you guys have been working as researchers. You've been working with this kind of data using firstly traditional machine learning models, like the traditional ones you were mentioning, and then now more with the deep learning ones. And it's I'm sure you can I'm sure you can reinforce this, but they are extremely valuable. It's not just AI, as in people might think about with large language models. Like there is so much value, and having that alongside those traditional models as well.
Yes, exactly. And I think where they mostly, like where they actually really help the most is exactly this variability problem. And they
Yeah,
understand the context, because that's the problem often in machine learning models. So if you do, for example, if you use pixel classifiers that are very often intensity based and so on, you can get somewhere. You can get maybe 80%, maybe. So for example, we actually use these models to generate first segmentations that we then refine. We use more machine learning or classical methods to do like the first segmentation that we then refine manually, that then will go into the AI model. But what these deep learning or models are really good at doing is understanding the context and learning the context, probably even where in the brain certain signal occurs, because there are areas where certain cells are clustered tighter than in other areas. Even though it's the same staining, but you might have very different types or like how the signal pattern looks like depending on where you are in the brain. And I think this is a context that deep learning models can understand, and this is extremely helpful. And also just this variance, you can teach that certain, even though for example, different artifacts that you have in your brain scans, there will always be artifacts, always. And they're actually even different for every machine, which makes it even harder. And you might not sometimes know, or there are new ones sometimes coming. And I think that these deep learning models can, by knowing what the standard is or how usually things look like or what you're searching for, it can ignore or in a way try to handle it. Obviously, the biggest problem is if you actually have under the artifact a really bad, like you have a signal and you would like to measure it. This is very difficult situation. But I even think that it's possible to teach the models to detect the signal inside the artifact area, just maybe being very conservative if it's blurry or depending on what the artifact is. So there, it definitely helps. So the variability of the signal, I think this is the biggest point here.
And a question on your approach to using AI or deep learning models at Vibraint. Like, how important are these models for your analysis, and what is your approach to them sort of moving forward? Where do you guys want to sit within the space? Is AI a speciality that you're looking to focus more on?
Definitely, actually. I mean, because we would like to help people who image using different microscopes, our aim is to also build deep learning models that are able to segment data from many different modalities. And this is ongoing work and it will take some time. I think for some more standard experiments, like for example, c-Fos for studying brain activity, it is possible to create models, and just keeping every time in mind that doing a QC, refining. I think it is possible actually to create models that can generalize for different imaging machines. So I think it's a big part of what we focus on. We haven't touched that much right now on registration, the aligning samples to atlases. Right now, we use a tool called elastix for this. Over the years, it's actually quite the old tool, but over the years, there have been many, many publications coming out with new ideas how to do it, because you need to non-linearly deform the brain sample to fit an atlas, or then atlas to fit the sample. And you can do it only in certain limits before things start going bad, let's put it that way. So registration is another challenge. It's actually also a very big challenge. And there have been also deep learning models that try to do that. I have every time tried them out, and I have every time come to the conclusion that they are either worse or as good as the model or the tool that I have been using until now. However, I do think that in that space, this registration challenge, deep learning will have a very big role to play. And I think there are possibilities to first of all speed it up, but also to make it more accurate. And it's more about designing how this whole pipeline should be, or how the models should be trained or what the model should be. I don't say that we are the experts in creating AI architectures. Hopefully in the future, we can also do that. But right now, it's a lot about applying different algorithms and techniques and deep learning models that we know that work. But it's registration is an interesting topic itself. It's one of the biggest challenges also, because you need to, for example, if you use a certain tissue clearing protocol or tissue processing protocol in general, your brain samples, the brain scans in the end look different. And even though we use a light sheet brain template it's based on a certain, it's made of brains that come from a certain tissue processing protocol. So we know that it works for that protocol. It doesn't necessarily perfectly work for all the other protocols. So it might be that we need to find ways to handle these samples that come from other protocols. One way of doing it is right now to create new brain templates, which should then make a big effort to align these templates with each other. So the different atlases with each other, because then you have like transformation chain, so to say, that you take a brain sample from a protocol, map it to the template of the protocol, which then connects to, let's say, Allen Brain Atlas and so on. So there are ways to circumvent the problem, but they are not efficient, so to say.
And is that, at what level do those sort of custom atlases come into play? Is that like at a client level, at like a workflow level, where there's specific markers, equipment and processing being used, so you apply it to like groups or like batches where people are doing similar things? Where does it fit into that?
I think so there are many different tissue clearing protocols out there, and I think labs tend to have one of them that suits best their needs, and but sometimes it's also the marker, or so to say, for example, if you do in situ hybridization protocols, we also do them in-house, and they definitely need their own brain template also, because the samples look a lot different compared to the standard iDisco protocol, for example. And so we need to handle these issues. We need to find ways. And obviously the easiest would be if you have a registration tool that can map an atlas, or can map a sample to an whatever atlas. But right now, a tool like that does not exist. And there are obviously also other challenges. Is, for example, sometimes if samples are not perfectly intact, there are some pieces missing, or if there are cuts in the brain. So these things all affect the registration quality. And even, it's even how far a brainstem is cut away, because then brain deforms in a certain way where you have smaller or bigger void areas, so to say. So deformations are different, and it's actually consistency is important, and these standard protocols are very important, and I think it's also very important to share them.
Yeah, that's fascinating, and again, it all just goes back to this consistency, quality control that you've mentioned so many times, because especially when, like, part of the value in creating these data sets is being able to compare them, or, well, reproduce them firstly, being able to reproduce the results, but also being able to compare them to other studies and other results. That's really important. So yeah, I think that's really interesting. And even just to hear, like, there's so many parameters so many tunable parameters that can differ between any given study, but between two separate brains, for example. So yeah, it's very insightful. So moving on from that, we've been talking about the quality control, working with different microscopes and different systems, what is your ability to work with these different systems and this variability between tools and analysis pipelines?
Our image analysis pipeline is also built very modularly, and it's very flexible. And I believe it is possible to apply that for many different imaging protocols. So tissue processing, but also image acquisition protocols. So there are many variables that can be adapted to a certain data set, definitely. But I also would like to emphasize that the conversation of, before actually really starting up an experiment, if you want to quantitatively analyze this, what comes out of a microscope, it's definitely very helpful to have consultancy sessions before, to be sure that every step actually provides the optimal results. That would be always the most wished setting, to have a close collaboration, and then there is always a way.
No, and that leads on quite nicely as well to ask about some examples of the kind of signals that you've analyzed, the things that you've worked with. So I wonder if you could touch on that a bit.
Our standards, so there are a few standard experiments that we do very often. We analyze very often, we also image very often brain activity, where we use c-Fos as a proxy. So we quantify then counts and densities of c-Fos positive cells. That we have been doing a long time. It works wonderfully. One of the examples is also biodistribution, meaning that, for example, if you want to, let's say you have compounds that you would like to know where these drugs are in the brain, where are they localized, then we measure signal intensities of these fluorescent labeled compounds in the brains. That's also very standard. Different markers of different cells, for example, is typical. For example, TH staining in Parkinson's studies, or we have also worked on quantifying plaques in the brain, or even very often there's also the studies where viral tracing is studied in the brain, and so basically reconstructing tracks in the brain and understanding the spread of either anterograde or retrograde viruses. So many different things. Also, neuroinflammation is, so we have studied microglia, which is very, very fun, I need to say.
Yeah,
so this is also one of the pipelines that we have. So I would say that it very often obviously depends where the staining we have gotten staining to work. But nuclear stainings are usually very easy. c-Fos is one of them, also p-STAT3 and so on. But cytoplasmic ones are sometimes more tricky. Like I mentioned TH before. This is more tricky because you also have the processes that then it's a little bit harder to detect single cells, and then you need to have techniques to do that that we have. And yeah, so all these, they come with their own dynamics, and we have then separate pipelines to analyze them.
And what are some of what are the different endpoints there? Because there's some really interesting study pathways there. But what are the endpoints that you get out of this, or you can produce?
Yes, so I would say that, I mean, count seems to be always the thing that the people ask for. Is also a very standard thing. So now we look into, so we always have counts and densities as one of the endpoints. Then we have started using coverage of a signal. So basically ratio in the brain region, how big area of this brain region is covered with the signal. And then also intensities, where you can look at either you can accumulate intensities in brain region, or you can look at means or medians and so on. So these are very typical ones that we use. There is also possibility, this is a little bit more complex, but for example, analyzing how far, for example, certain objects are from others. So for example, how far could objects be from blood vessels, or so there's comes also these kind of metrics into play. Yeah.
And do you have a standard set of those, or can it be bespokely built for each project?
So these are actually standard, and they are fully part of the pipeline and implemented. Yeah. And then it's more also about understanding if maybe for certain biological questions, new metrics need to be implemented even further.
Can you give an example of where an obvious metric might be difficult to obtain, or even be a wrong choice?
Yes, definitely. One of the interesting, also challenging projects has been the microglia quantification, because microglia, you can, I mean, it's very intuitive to say, let's count them. And interestingly, in most of cases, it's possible. But the problem is that where you have inflammation, they actually tend to clump together. So they tend to migrate together, and they also change their shape, which actually in the end, it is very, very difficult to count them. And we have sometimes made the effort and really, put all there to count every single cell, and then understood that, the count is actually not changing in the brain region. So it's also a lot about density, local densities. That's also maybe where the voxel-wise analysis becomes very important, because sometimes within the brain region that's atlas defined, you will think that there are differences between groups, but actually counts might not change at all.
Interesting.
However, local densities might change a lot. And there, for example, analyzing coverage or biodistribution would be a little bit more relevant, because usually we haven't seen differences in different experiments in counts.
That is really interesting. That's a great example. And then moving on to, you've already touched on this a bit about registration, but how reliable is it? And you've touched on some of the things that can impact the registration in terms of like the methodology that's used. But what can you do to sort of circumvent that?
Exactly. The registration usually is very reliable when you use the same brain template from brains that have been processed the same way from the same protocol. This is, I would say, very reliable. Now, when we start talking about, there are obviously different. So the brain template that we use is made of male mice, for example. This also means that there might be changes when you would use female mice, or if you use older or younger mice. The same goes for different strains. There are differences. There are differences between different strains of the same species. So these things need to be kept in mind. And sometimes it is maybe the area that you're interested in your experiment. It is this one area is mapping okay, but there might be other areas that are mapping less good. Then this might be okay. But this all needs to be evaluated, and for that we have automated quality control for these kind of things. But it's also important for us then to know where we would expect the signal, so that the quality control can be can be done optimally. So basically, different sexes, different ages, and different strains, and then different protocols where the tissue processing has gone through, they all definitely affect the registration and need to be taken into account. Yeah.
Thank you. And to switch things up a bit now, I'd like to talk about CNS-Voyager, your platform. I've spoken to some of your other team members about this, and it's a really exciting platform. So can you just give me for people who aren't familiar, can you give an overview of what CNS-Voyager is and how it could enable people to interact with their data?
Yes, definitely. So our brain viewer is, the idea of this is to be able to visualize and explore complex signals in 3D, but also in 2D. And as I mentioned before, it is because brain is so complex and it is a three-dimensional organ, and the data we acquire is three-dimensional. It's very, very important to also present and explore the data in 3D to see all the whole spatial signals at once and understand what is going on. So we have a 3D viewer, but also 2D view where it's possible to go through the brain and different either coronal, sagittal, or horizontal views. And the idea is that when we perform our, for example, contract research and we have performed image analysis, then all the results that we have gathered, they will be uploaded to the CNS-Voyager. All the brain maps will be available in three-dimensional atlas space, where you can, for example, pinpoint interesting areas for you, and you even get the in vivo coordinates of the signal there. So you have a way of interacting with your own data and it comfortably and easily, because that's usually the problem. You need to open thousands of images or thousands of plots, and this is just becomes a pain and you don't know what to look at. So the idea is that also, for example, all the metrics that we have extracted, that we are able to provide an unbiased view of all the measurements that were done without, first you have an unbiased view, and then there is also a way to pinpoint something that might be interesting. Then the whole idea of the viewer is to be able to integrate different experiments and different data sets, so even from different modalities. Because for a long time, we have been doing brain activity experiments, for example, and said, oh, wonderful, we know now where the brain is activated by giving that drug. But then you start asking, but what does it mean, really? And then you need to complement this information with a different technique. So, for example, gene expression profiles, to understand what kind of cell types they really are. So you can start doing more targeted analysis, or you can do several experiments. Maybe they are totally separate experiments with different paradigms and so on. You can combine all this information in one platform to have your own portfolio and start comparing different brain maps with each other.
That is, I can only imagine how sort of transformational that is as a workflow from opening, working with that data so manually and being able to compound the analysis from different modalities or different studies into one space, and the kind of questions that you can answer there. It's quite an exciting sort of vector for science as well, to be able to elevate data analysis and visualization in that kind of way, because it's just such a more natural way of engaging with the data. And I mean, you don't even need to sort of touch on why there's an advantage to that over trying to do it locally on your computer. It's faster and way more insightful by the sounds of it.
Yes, we see that as a guiding tool for the whole experimental process, and not only having one experiment that you do, but maybe you use it for first because we will have public data sets available also on the platform where you can use these data sets to generate hypotheses that you can then perform a study, again, have it then in the viewer, maybe find certain cell types that you find interesting or area that you want to go more in depth, or maybe even surgically target these cells. So you can do follow-up experiments, and it kind of all feeds. So it's an experimental tool. It becomes part of neuroscientist toolbox. Yeah, that's the aim.
Yeah, and it's giving you access, like, as well here to other data, to other studies that you can use to inform like your decision making going forward. That's really interesting. Thank you for sharing some insight on that. So ultimately, who is the Lightsheet Journey image analysis module for?
I think this module is actually for academics in universities, or also biotech and pharma companies who have access already to a light sheet microscope, and either maybe they're doing already some experiments, they have a laboratory, they are able to do tissue processing, and they're good at imaging samples. And we are happy to support them with the image analysis part. Exactly, it's meant maybe for the groups that don't necessarily have a computer scientist accessible for them, or computer scientist that is who has the very specific knowledge about brain image analysis.
And what are the sort of scales on that spectrum that you will work with, to like the smallest groups or sample sizes to the largest? What range do you sort of operate within there?
We are very flexible, to be honest. To mention the standard studies, usually, this is now from the statistical analysis perspective, we would always recommend to have minimum 8 animals in a group. We have also sometimes done it with 6 animals in a group, but this is just a limitation that needs to be, noted, that exists when people use less animals in a group. But I wouldn't say that there is a limitation, lower or upper bound, so to say. Yeah.
Good to know. What data formats can be analyzed, and how should the data be shared?
Nowadays, the microscopes, light sheet microscopes, usually output both TIFF and H5 formats. I'm not sure if Zarr is already possible output, but basically these kinds of data formats work very well in our pipeline, and the data will be shared via cloud bucket that we will create for our customers, and we also give them guidelines how to upload the data sets, because they might also be quite big in some cases. So everything happens via cloud, basically. Yeah.
And what do you need from customers upfront to scope out a project?
We would really like to first have a meeting and understand what is the experiment you do, what are the experimental questions, so that we can find a good metric to evaluate. And that also means coming back to the important point, that it would be beneficial to, before doing even imaging, to consult with us, so that by knowing what is your experimental question, what have you thought about metrics you would like to analyze and quantify, that we can find optimal settings before you get going with scanning.
Excellent. And do you provide consulting if researchers would like to analyze their own data in-house.
Exactly. We do also that because maybe you have very special stainings, or you would really like to learn yourself how to analyze the data. There are also open source tools available. For example, BrainGlobe has very good open source tools. And I think that we are happy to consult for, for everyone who would like to get on this journey of image analysis, and maybe, even discussing ways how to do further analysis. There are many further techniques that can be done after you have extracted actually your metrics. And so I think we are very open and are very excited about maybe even finding new ways how to analyze these data sets together. Yeah.
Can you touch on a couple of examples of some of that further analysis that you can do?
It's, for example, possible to do, so to say, functional connectivity analysis if you measure brain activity, for example, or even graph-based analysis. So there are starting to, because these are all that go into question, okay, what now when I have done my first experiment? How can I further use the data to get even more information out of it? And they are not necessarily always standard techniques, because or standard in every setting or every pipeline. So it comes with a scientific question that's behind it, and then how to get there. Yeah.
Perfect. Well, Johanna, thank you very much for sharing so much of your knowledge about what you're doing at Vibraint and the LS journey.
Thank you so much, Harry. Also, it was my pleasure to have the interview.